Okay, welcome to the deep dive. Have you ever really stopped to think about that invisible symphony, the one constantly playing inside your brain? Or, how just a tiny, carefully placed chemical can completely shift your mood, or silence pain, or even like sculpt your actual memories? Today, we're embarking on a journey, a deep one, into a realm that's fascinated humanity basically forever. The astonishing world of drugs acting on the central nervous system. Our blueprint for this expedition, our stack of sources, comes from the incredibly dense but utterly foundational KATZUNG's Basic and Clinical Pharmacology 16th edition. Specifically, that pivotal chapter on CNS drugs. Our mission really for you is to transform these intricate pages of neuroscience of pharmacology into accessible, truly impactful insights. We want to arm you with the kind of deep foundational understanding, the kind of mastery, frankly, that feels like you're one of the elite professionals in this field. We're going to peel back every layer, scrutinize every concept, explore every mechanism to really grasp how these impossibly complex systems orchestrate our very existence.
It's quite remarkable, isn't it, when you think about it, how CNS drugs, which, date back to some of the earliest discoveries by primitive humans, they continue to be among the most widely used pharmacological agents in medicine today. It's incredible. Their influence is just profound, ranging from, life-altering treatments for severe neurological and psychiatric conditions, right down to simply enhancing our daily well-being. Think about something as fundamental as relieving pain or suppressing nausea or even just reducing a fever. For centuries, honestly, our understanding of how these drugs actually work was frustratingly vague, largely because of the sheer daunting complexity of the brain itself. But in recent decades, we've witnessed dramatic, almost revolutionary advances in methodology. We've moved from these sort of crude observations to being able to study drug action with breathtaking precision, down to individual neurons, even single receptors nestled within synapses.
That's more than just a leap, isn't it? It sounds like a complete paradigm shift, moving from just a general idea to pinpointing the exact molecular choreography.
Precisely, yeah. And this newfound precision has unlocked several major developments in how we comprehend the CNS and how drugs interact with it. First and foremost, it's now, unequivocally clear that nearly all drugs that exert effects on the central nervous system, they do so by acting on specific receptors. And by doing that, they modulate synaptic transmission. Now, while a few agents like general anesthetics or maybe even alcohol might initially seem to have rather non-specific actions, on cell membranes, even these ultimately lead to demonstrable alterations in synaptic transmission. So the core principle really holds true. The brain's communication pathways are the primary targets.
Okay, so even the things we might think of as like the blunt instruments, they're still incredibly sophisticated in how they ultimately impact those communication pathways. That feels like a critical distinction for true mastery.
Absolutely, it is. The second major development is that the drugs themselves have become indispensable tools, really invaluable for the scientific study of CNS function. They allow us to probe and understand everything from the intricate, kind of uncontrolled electrical activities seen in convulsions to the really elegant mechanisms by which long-term memories are formed and consolidated. Both agonists, those are compounds that mimic or enhance the action of natural neurotransmitters, and often, strikingly, are even more selective than the endogenous substances.
More selective than the body's own chemicals. Wow.
Sometimes, yes, and antagonists, which block receptor function, are both invaluable in these research endeavors. They essentially give scientists the ability to selectively, turn on or turn off specific brain processes, or to precisely mimic particular brain chemicals to observe their downstream effects.
It's like having these hyper-specific molecular precision tools that let us functionally dissect the brain without ever needing a scalpel. That's a powerful approach, yeah.
And third, the detailed unraveling of the actions of drugs that have known clinical efficacy. This has provided some of the most fruitful hypotheses regarding the underlying mechanisms of various neurological and psychiatric diseases. Take, for instance, the profound insights we've gained into how antipsychotic drugs interact with dopamine receptors. This detailed pharmacological knowledge has literally provided the very basis for our current understanding of the pathophysiology of schizophrenia. Similarly, extensive studies focusing on the effects of various agonists and antagonists on GABA receptors, gamma-aminobutyric acid.
Right, GABA, the main inhibitory one.
Exactly. Those studies have paved the way for entirely new concepts about the pathophysiology of conditions like anxiety and epilepsy. So the drugs we use aren't just treatments, they are invaluable clues that guide our understanding of the very diseases they aim to ameliorate.
That's a powerful cycle, isn't it? A self-reinforcing loop.
The drugs illuminate the disease, and that deeper understanding in turn informs the design of even better, more targeted drugs. That's real scientific advancement.
Indeed, it is. And to truly appreciate the nuanced effects of any drug on the central nervous system, you really have to adopt a multi-level perspective. You need to understand the brain's organization right from its smallest components, the genes and molecules, all the way up through complex neural circuits, and ultimately how those circuits manifest as behavior. So this deep dive will systematically introduce you to this functional organization and the diverse synaptic transmitters that form its basis, so you can truly master how these drugs exert their precise and often profound effects.
Okay, so to begin that journey toward mastery, before we even talk about how drugs act, we really need to grasp the incredibly intricate system they operate upon. We're talking about the central nervous system, the CNS, composed of the brain and spinal cord. This is the ultimate command center, right? The master integrator of all sensory information constantly flooding in, the meticulous generator of our motor output, and the grand orchestrator of all the complex behaviors essential for us to interact with our environment and, well, quite literally for our species' survival. And when we say intricate, we mean it on a scale that's almost unfathomable. The human brain alone, for instance, contains an estimated 100 billion interconnected neurons.
That number, 100 billion. It's truly staggering, difficult to even visualize. And these neurons aren't just floating around chaotically, no. They are meticulously organized and surrounded by an equally vast network of various supporting cells called glia. Throughout the CNS, neurons are either clustered into distinct groups, which we call nuclei, or they're arranged in highly organized layered structures like you see in the cerebellum or the hippocampus. The precise connections among these neurons, both within and between these clusters, form the incredibly complex circuitry that meticulously regulates the flow of information throughout the entire CNS. It's not just a collection of cells, it's a beautifully organized, hierarchical, and highly distributed processing network, a true marvel of biological engineering.
So if we zoom out for a second,
Yeah.
and connect this to the bigger picture of brain function. Neurons are the fundamental units of information processing. They're these remarkable electrically excitable cells that not only process information, but also transmit it with astonishing speed and precision. And they use an electrochemical process, right?
Exactly, both electrical signals and chemical messengers. We classified the many types of neurons in multiple ways, by their location in the brain, by their specific function within a circuit, and crucially for our purposes today, by the type of neurotransmitter they release. This specific classification is absolutely vital for understanding how drugs can precisely target particular neuronal functions.
Okay, so a typical neuron, maybe for those visualizing, what does it consist of?
Right. A typical neuron consists of a central cell body, which we call the soma, and then specialized extensions protruding from it, the dendrites and the axon. You can see this structure laid out nicely in figure 21:1 in KATZUNG's. The dendrites. They often form these incredibly branched and complex dendritic trees. They're essentially the neuron's sophisticated reception antennas, designed to receive and integrate a multitude of incoming electrical and chemical inputs from other neurons. Conducting this processed information inward towards the cell body, imagine an exquisitely sensitive, highly branched antenna array constantly picking up and filtering signals.
And then the axon. That's the neuron's single dedicated output signal carrier, right? Like the main cable sending the message out.
Precisely. It extends, sometimes over surprisingly long distances, from the cell body, and typically branches out distally to make contact with multiple target neurons or cells. You can think of it as the brain's high-speed fiber optic cable, efficiently sending its precisely calculated signals out.
Okay, and at the very end of that axon, that's where the synapse happens.
Exactly. At the very end, you have the axon terminal. This terminal forms highly specialized junctions with other neurons. Those are the synapses. It is at these synapses that the electrical signal arriving at the terminal gets converted into a chemical signal. Neurotransmitter chemicals are released, and these chemicals then precisely interact with specific receptors located on the membrane of the receiving or postsynaptic neuron, and that completes the communication circuit.
Now, here's where it gets really interesting, I think. Often overlooked beyond the star players, the neurons, there exists this entire vast army of non-neuronal support cells, collectively called glia. These cells perform a remarkable variety of absolutely essential functions, often operating quietly in the background, but without them, the central nervous system simply could not function. They're kind of the indispensable stage crew, right? Making sure the neuronal show runs flawlessly.
That's a great way to put it. And among these crucial glial cells, astrocytes really stand out. They are the most abundant cell type in the brain. They play pivotal homeostatic support roles, serving as crucial intermediaries. For instance, they provide vital metabolic nutrients to neurons, making sure they have the energy to function. They also meticulously help maintain the critical extracellular ion concentrations, which is absolutely vital for proper neuronal firing and signaling. But what's truly fascinating is how intimately astrocyte processes are associated with neuronal synapses. They're not just providing broad support, they're actively involved in the rapid removal and precise recycling of neurotransmitters right after they're released into the synaptic cleft, essentially cleaning up the chemical messengers to ensure signal precision.
So they're not just passive support structures at all. It sounds like they're deeply involved. Active participants.
Increasingly so, yes. They play these recognized active roles in dynamically regulating synapse formation and function. For example, astrocytes express their own array of neurotransmitter receptors, allowing them to detect neuronal activity. And even more surprisingly, they can release their own neuromodulators that can directly alter neuronal activity. So they're not just quiet background players, they're truly active participants in the brain's incredibly complex information processing, contributing directly.
Okay, what about the other types of glia? Oligodendrocytes, for instance.
Right, then we have oligodendrocytes. These cells are the central nervous system's functional equivalent of Schwann cells, which serve a similar role out in the peripheral nervous system. Their primary and utterly critical function is to wrap around the axons of projection neurons, forming a specialized insulating layer, the myelin sheath. This myelin sheath is fundamental. It acts like electrical insulation around a wire, dramatically increasing the speed at which action potentials the neuron's electrical signals can propagate down the axon.
So it's like boosting the internet speed of the brain's network. Makes sense.
Exactly. Faster communication.
And this insulation isn't just about speed, right? It's profoundly important clinically. I know damage here is key in MS.
Absolutely. Damage to these oligodendrocytes, and thus to the myelin sheath, is a characteristic hallmark of multiple sclerosis, a devastating neurodegenerative disease. This makes these cells and the processes of myelination a key target for drug discovery efforts aimed at treating these demyelinating diseases. Understanding their function is critical to unlocking new therapies.
Okay, and the third main type, microglia, what's their role?
We also have microglia. These are highly specialized macrophages actually derived from the bone marrow. They migrate into the CNS during development and establish themselves as the brain's primary immune defense system. They become actively involved in neuroinflammatory processes in many pathological states, including major neurodegenerative diseases like Alzheimer's and Parkinson's. However, beyond disease, their roles in normal brain function are increasingly being appreciated. For instance, microglia engage in a process called synaptic pruning.
Synaptic pruning, what does that mean exactly?
They literally engulf and eliminate synapses that are no longer needed or are perhaps dysfunctional.
Yeah.
This process is absolutely crucial for normal circuit development, allowing the brain to refine its connections as it learns and grows.
So they're like the brain's sophisticated waste management and recycling crew, but highly selective and dynamic. And this pruning, it sounds like it could be a double-edged sword, couldn't it? Beneficial, but maybe also detrimental.
It absolutely is, yes. While essential for healthy brain development and plasticity, excessive or dysregulated microglia-mediated synapse elimination has been strongly linked to the progression of conditions like schizophrenia and Alzheimer's disease. This really highlights their complex and critical roles, capable of both maintaining brain health and contributing to pathology when their delicate balance is disrupted. For anyone aiming for mastery, understanding this dual nature is key. Now, this intricate system of neurons and glia is also remarkably protected, which raises an important question. How does anything, especially a drug, get into this incredibly delicate and shielded environment, the brain? The answer lies in the blood-brain barrier, or BBB. This isn't just a physical wall, it's a protective, functional separation of the circulating blood from the extracellular fluid of the CNS. It acts as a highly selective gatekeeper, rigorously limiting the penetration of substances, including many pharmacological agents, from the bloodstream into the brain tissue itself.
So it's like the brain's exclusive VIP entrance. Extremely strict bouncer, very selective guest list, making sure only what's absolutely necessary gets in.
That's a perfect analogy, yes. This selective separation is achieved by the presence of exceptionally tight junctions between the capillary endothelial cells lining the brain's blood vessels. These junctions are so tight that substances can't simply squeeze between the cells, unlike in other parts of the body. Furthermore, these tight junctions are reinforced by a surrounding layer of astrocyte end feet, those extensions from the glial cells we just discussed. Because of this formidable barrier, for drugs to successfully enter the CNS, they must either be highly hydrophobic, meaning lipid-soluble, able to pass directly through the cell membranes.
Right, dissolve in fat, basically.
Exactly. Or they must engage specific active transport mechanisms designed to carry them across.
That immediately eliminates why drug design is so critical when developing any medication meant to affect the CNS. You could have the most brilliant compound, but if it can't cross that barrier, it's useless, right?
Precisely. Consider second-generation antihistamines, for example. Older ones famously cause significant drowsiness, but newer ones, like loratadine, were specifically developed to be significantly more polar. This increased polarity drastically limits their ability to cross the BBB, so they primarily act on histamine receptors in the periphery, reducing allergy symptoms with far less central sedation. On the other hand, essential nutrients like glucose and amino acids must get into the brain. They achieve this because the BBB is equipped with specific, dedicated transporters designed to ferry them across.
And this also perfectly explains the classic example of L-dopio versus dopamine for Parkinson's disease, doesn't it? That's like the textbook example of the barrier's impact.
It truly is, yeah. L-DOP, which is a metabolic precursor to the neurotransmitter dopamine, can successfully enter the brain by utilizing an amino acid transporter at the BBB. Dopamine itself, however, cannot cross the BBB directly. It lacks such a transport system and isn't sufficiently lipid-soluble. Therefore, in Parkinson's treatment, orally administered L-DOP is used to boost CNS dopamine levels because it's the molecule that can penetrate the brain, where it's then converted to dopamine. It's a clever biological workaround.
A very clever workaround. So it's a sophisticated gate, but are there any specific regions of the brain that naturally lack this strict VIP entrance? Any exceptions to the rule?
Yes, there are, and they're known as the circumventricular organs. These are specific, specialized regions that naturally lack a typical blood-brain barrier. They are strategically located where the brain needs direct and rapid access to substances circulating in the blood. Examples include areas that need to sample the blood for regulatory purposes, like the area postrema, often called the brain's vomiting center. It directly samples blood for toxins to trigger nausea or vomiting.
Oh, makes sense. So it needs direct access to detect potential threats.
Exactly. Other circumventricular organs are regions that secrete neurohormones directly into the bloodstream. These specialized functions necessitate direct access, hence the absence of the barrier in those specific spots.
Okay, here's where it gets really captivating, I think. Showing how nature, through millions of years of evolution, has proven to be the most brilliant chemist. Evolution has produced this incredible array of natural toxins, and remarkably, pharmacologists have leveraged these very toxins as invaluable tools for precisely characterizing ion channels, which are fundamental to all brain function. When you think about it, nature's been running sophisticated chemical experiments for eons.
Indeed. The sheer diversity is astonishing. Consider the predatory marine cone snails. Hundreds of different species, each one uniquely kills or paralyzes prey using venom containing 50 to 200 distinct peptides. What's even more remarkable is the minimal duplication of peptides among species, meaning there's an enormous, virtually untapped library of unique compounds out there. Other animals like snakes, frogs, spiders, bees, wasps, scorpions. They similarly produce incredibly useful toxins, and of course, various plants have their own rich repertoire.
And what makes these natural toxins so exceptionally valuable for research into brain function and drug discovery? Why are they so useful?
Because many of these toxins act with exquisite specificity on ion channels. They provide an unparalleled wealth of highly potent and selective chemical tools for probing channel function. In fact, much of our current detailed understanding of the precise properties and mechanisms of ion channels comes directly from studies using only a small percentage of the potent and selective toxins available to us. Many typically target voltage-sensitive ion channels, crucial for electrical signaling. But a significant number also block ligand-gated ion channel receptors, vital for chemical signaling.
Can you give us a couple of particularly memorable examples, ones that really highlight their utility?
Certainly. Take tetrodotoxin, TTX, famously from the pufferfish. This toxin potently and selectively blocks voltage-gated sodium channels from the outside of the neuron. This is why, if ingested, you'd experience severe numbness or paralysis. The fundamental electrical signals for nerve impulses are completely blocked.
Okay, that's a clear link.
Then there's alpha-bungarotoxin for marine snakes. A potent toxin, it's an irreversible antagonist at nicotinic acetylcholine receptors. It binds so tightly it essentially disables them. This characteristic makes it a classic and irreplaceable research tool for studying those specific receptors. And linking back to our earlier GABA discussion, picrotoxin from a Southeast Asian vine specifically blocks GABA receptors. This blockade causes widespread convulsions, providing a vivid, direct demonstration of how inhibiting an inhibitory system leads to uncontrolled, generalized excitation, profoundly illustrating that delicate balance in the brain. So, to connect this to the bigger picture, how neurons actually come alive and communicate, their dynamic function really hinges on two primary types of ion channels embedded in their membranes. These channels are fundamentally defined by how they open and close, or how they are gated. Understanding these two gating mechanisms is crucial for appreciating how drugs operate.
Okay, so what controls that gating mechanism for the first type? What makes them open or close?
One type is the voltage-gated channel. As the name suggests, these channels respond directly and rapidly to changes in the cell's membrane potential, that electrical charge difference across the neuronal membrane. A quintessential example is the voltage-gated sodium channel, found in high concentrations at the axon initial segment, the very beginning of the axon. This channel is absolutely crucial for initiating and then rapidly propagating the all-or-nothing fast action potential, the neuron's primary electrical signal down the entire length of the axon.
So a precise change in electrical charge across the membrane literally acts as a switch, opening or closing these channels and kicking off that incredibly rapid signal cascade.
Exactly, yeah. Beyond these rapid sodium channels, neurons also possess many different types of voltage-gated calcium and potassium channels. These are found distributed on the neuron's cell body, dendrites, and the axon initial segment. Unlike the fast sodium channels, these calcium and potassium channels generally operate on a slower timescale, and they play a crucial modulatory role in regulating the rate and pattern at which the neuron discharges action potentials. For instance, certain types of potassium channels open in response to sustained depolarization, effectively acting like a brake to slow down further depolarization and limit the number of action potentials a neuron can fire, precisely tuning its activity.
Okay, now let's unpack the next critical step. Once that electrical signal, the action potential, reaches the synapse, it undergoes this remarkable transformation, right? Switching from an electrical language to a chemical one. And that's precisely where neurotransmitter receptors come into play. And you said there are two fundamentally distinct classes, each with its own unique speed and style.
That's right. The first class are the ligand-gated channels, also commonly known as ionotropic receptors. These receptors aren't just single proteins, they are complex structures made up of multiple protein subunits that come together to form a channel that is an integral part of the receptor itself. When a neurotransmitter, which is the ligand here, binds to this receptor, it directly causes a conformational change that opens the ion channel.
So it's a direct, immediate action, like putting a specific key in a lock and instantly opening a door. No intermediary step needed.
Precisely. This direct binding typically results in a very brief opening of the channel, usually lasting only a few milliseconds to, at most, maybe tens of milliseconds. These ligand-gated channels are fundamental for mediating the rapid, fast synaptic transmission that we observe in the brain's precise hierarchical projection pathways. You can indeed think of it as a quick, direct phone call from one neuron to another, essential for immediate sensory processing and motor commands.
Okay, direct and fast. And what about the second class? How do they operate differently?
The second class are the metabotropic receptors. These are predominantly 7 transmembrane G-protein coupled receptors, or GPCRs, a vast and diverse family similar to those found throughout the body. What's crucial to understand here is that neurotransmitter binding to this type of receptor does not directly open an ion channel. Instead, the binding event activates an associated G protein, which then initiates the production of second messengers within the cell. These second messengers, in turn, mediate complex intracellular signaling cascades, setting off a chain reaction or domino effect inside the neuron.
Ah, okay, so if ionotropic receptors are the direct phone call, these metabotropic ones are more like setting off an internal chain reaction, a domino cascade inside the cell, leading to more prolonged effects.
That's an excellent analogy. Yes, it captures their essence. This cascade mechanism allows for much more complex and often more prolonged effects within the neuron. In neurons, activation of metabotropic neurotransmitter receptors frequently leads to the modulation of voltage-gated channels, bringing us back to our earlier discussion about electrical excitability. These interactions can occur through two principal mechanisms. First, via membrane-delimited pathways, where the G protein itself, often its beta-gamma subunits, directly interacts with a voltage-gated ion channel within the membrane.
Can you give a specific example of that direct membrane-delimited interaction? How does that play out?
Certainly. When these G proteins interact with presynaptic calcium channels, they directly inhibit their function. This is a key mechanism for the inhibition of neurotransmitter release that happens when presynaptic metabotropic receptors are activated. It essentially puts a very precise brake on the release of further neurotransmitters from the sending neuron. Conversely, when these receptors are located postsynaptically, their G proteins can activate or cause the opening of potassium channels, which results in a slow, sustained postsynaptic inhibition.
Okay, that's one way. And the second primary way these metabotropic receptors modulate voltage-gated channels.
The second way is through the generation of diffusible second messengers. In this scenario, the activated G protein triggers an enzyme, for example, adenyl cyclase, which then synthesizes a diffusible second messenger like cyclic AMP or CAMP. These messengers aren't confined to the membrane. They can then act over considerable distances within the cytoplasm of the cell, leading to more widespread and generally more prolonged effects. A classic example is how beta-adrenergic receptors, when activated, generate HAMP, influencing a whole host of intracellular processes.
And because of this cascade nature, the speed and duration of the effects are vastly different from the direct ionotropic channels, aren't they? This seems critical for understanding their functional roles.
Absolutely. While ionotropic receptors produce very brief, transient effects, milliseconds usually, the effects of metabotropic receptor activation are considerably longer lasting, spanning from tens of seconds to minutes. This prolonged duration is precisely because the intricate intracellular signaling cascades they initiate. This longer time course makes metabotropic receptors predominant in the brain's diffuse modulatory neuronal systems, which we'll delve into shortly. They're primarily involved in orchestrating more global, sustained changes in overall brain states rather than rapid, specific information transfer. Now, this raises a crucial question for anyone pursuing mastery. How do all these distinct channels and receptors synergistically come together to enable the dynamic communication that defines brain function? Well, the vast majority of neuronal communication within the central nervous system occurs via chemical synapses. Think of them as incredibly precise, microscopic chemical messengers facilitating an intricate, beautifully choreographed dance of information transfer from one neuron to the next. While a few instances of direct electrical coupling between neurons exist, primarily involved in synchronizing neuronal discharge, they're generally not considered significant sites of drug action like chemical synapses are. For pharmacology, the chemical synapse is truly where the action is.
So the core of how our incredibly complex brain communicates, processes thoughts. It's essentially a series of rapid, intricate chemical handoffs. Let's walk through that precise process step by step, from electrical signal to chemical message and back again.
Okay. It all begins with an action potential, the neuron's fundamental electrical signal, propagating down the presynaptic axon until it arrives at the synaptic terminal. This electrical arrival then triggers the activation of specialized voltage-gated calcium channels embedded in the membrane of that presynaptic terminal. And it's an important detail to note. These specific calcium channels, indispensable for initiating neurotransmitter release, are generally resistant to common calcium channel blockers like verapamil that affect other parts of the body. However, they are exquisitely sensitive to certain marine toxins and specific metal ions, linking back to our discussion of natural toxins as research tools.
So very specific, highly regulated gates for calcium, opening only under precise electrical conditions. And once that gate opens, calcium rushes in, correct?
Precisely. As calcium ions flow into the presynaptic terminal, this rapid increase in intracellular calcium concentration acts as the pivotal signal promoting the fusion of synaptic vesicles. Those tiny membrane-bound sacs meticulously filled with neurotransmitters with the presynaptic membrane. This fusion event then culminates in the release of the neurotransmitter chemicals into the synaptic cleft, that minuscule gap between the sending and receiving neurons. This entire remarkably rapid release process accounts for the majority of the approximately 0.5 millisecond synaptic delay, the brief lag between action potential arrival and the postsynaptic response.
Okay, 0.5 milliseconds. That's incredibly fast. And once that burst of neurotransmitter is released into that tiny cleft, what happens next at the receiving neuron?
Once in the synaptic cleft, the neurotransmitter molecules rapidly diffuse across that minuscule space and bind to specific receptors located on the postsynaptic membrane of the receiving neuron. This binding event then triggers the opening of ion channels, either directly for ionotropic receptors or indirectly via second messenger cascades for metabotropic ones. This results in a brief, localized change in the postsynaptic cell's membrane conductance, its permeability to specific ions. This change in ion flow then dictates whether the postsynaptic neuron becomes more excited or inhibited, influencing the next stage of signal processing.
Okay, to truly master CNS pharmacology, let's unpack a fundamental concept. The brilliant way the CNS achieves its complex functions through this delicate balance of excitation and inhibition, the postsynaptic potentials. Those are how we precisely measure and understand this critical equilibrium, right? And this understanding really began with pioneering work by John Eccles and his team back in the 1950s.
That's right. Using intracellular recordings from spinal motor neurons, they were able to precisely measure a neuron's resting membrane potential, typically around -60 millivolts, a precise electrical baseline setting the stage for all activity.
Okay, -60 milliV, the baseline.
That resting potential is indeed the starting point. When an excitatory pathway connected to that motor neuron is stimulated, it produces a small, transient depolarization of the postsynaptic membrane. That's the excitatory postsynaptic potential, or EPSP. This depolarization happens because the excitatory neurotransmitter acts on its ionotropic receptor, causing a selective increase in the permeability of the membrane to positive ions, like sodium. This influx of positive charge brings the neuron's membrane potential closer to its firing threshold.
And crucially, these small individual EPSPs can actually add up, can't they?
Yeah.
This concept of summation is vital for understanding how a neuron decides whether or not to fire.
Absolutely. Two crucial types of summation determine whether an action potential is generated. First, spatial summation. If multiple excitatory synapses on a single neuron are activated simultaneously, their individual small EPSPs summate in a graded manner, leading to a larger overall depolarization. Second, there's temporal summation. If a single excitatory input fires repetitively and rapidly, its successive EPSPs can also summate over time, building upon each other. If these summed EPSPs, through either spatial or temporal summation, collectively reach a critical threshold potential, usually around -50 millivolts, then an all-or-none action potential is robustly generated and propagated down the axon.
So it's this magnificent integration system.
Yeah.
Multiple small nudges from different sources or rapid-fire nudges from a single source
Yeah.
can cumulatively push the neuron over that critical threshold. But then there's the equally vital other side of the coin. Inhibition. How does that work precisely?
Inhibition is absolutely indispensable for precise brain function. It prevents runaway excitation and sculpts signals. When an inhibitory pathway is stimulated, it causes a hyperpolarization of the postsynaptic membrane, meaning the inside of the cell becomes even more negative, moving away from the firing threshold. This hyperpolarization is achieved primarily by the selective opening of chloride Cl channels, producing an inhibitory postsynaptic potential, or IPSP. However, because the equilibrium potential for chloride, the point where chloride ions have no net driving force to move, is only slightly more negative than the resting potential, maybe around -65 millivolts. The hyperpolarization itself is actually quite small and contributes only modestly to the overall inhibitory action.
So wait, the slight hyperpolarization isn't the whole story, or even the main story for inhibition. What's the key inhibitory action then, if it's not just making the cell more negative?
You've hit on a critical point there, yes. The primary inhibitory action is what's elegantly termed the shunting effect. When the chloride channels open during an IPSP, they dramatically increase the membrane's conductance to chloride. This increased leakiness for chloride makes it much, much harder for the membrane potential to change significantly in response to any excitatory input. Imagine trying to fill a bucket that now has a large hole in its bottom.
Okay. So the excitatory current just leaks out before it can build up.
Exactly. An EPSP that would normally be strong enough to trigger an action potential might completely fail to do so during an IPSP because the inhibitory input effectively shorts out the excitatory current, preventing it from reaching threshold. It's a powerful and precise mechanism for dampening or even silencing unwanted signals.
That's a truly sophisticated way to dampen a signal or filter out noise. And is there a second distinct type of inhibition that drugs can target beyond this postsynaptic shunting?
There is, yes. And it's called presynaptic inhibition. In this scenario, unlike postsynaptic inhibition acting on the receiving neuron, neurotransmitter release itself is directly inhibited at the very same synapse. This can occur through what are known as autoreceptors, receptors on the presynaptic terminal that respond to the transmitter released by that same terminal, creating a local negative feedback loop. Alternatively, the released neurotransmitter can sometimes spill over from its synapse to affect neighboring synapses, also directly inhibiting their transmitter release. This adds another critical layer of control, acting directly at the source of the signal before it even crosses the synaptic cleft, offering yet another point of modulation for drugs. This raises a fundamental question for anyone truly aiming to master this field. Given the extraordinary complexity and dynamic interplay of synaptic transmission, where exactly do drugs fit into this intricate machinery? What's consistently fascinating here is that virtually all drugs acting in the central nervous system produce their effect by precisely modifying some specific step in this multi-stage chemical synaptic transmission process. These drug actions can broadly, but very importantly, be divided into two main categories. Those that act presynaptically, influencing the sending neuron, and those that act postsynaptically, affecting the receiving neuron.
Okay, so drugs are essentially finely tuned molecular keys or maybe specialized wrenches that fit into the intricate machinery of the synapse, subtly altering its function. Let's start by breaking down the presynaptic actions, things happening before the neurotransmitter is even released.
Indeed. Drugs can interfere with the very first step, the synthesis of neurotransmitters. Some drugs, for example, can block the specific enzymatic pathways necessary to create the transmitter molecules. Then, they can affect storage. Synaptic transmission can be significantly depressed by blocking the efficient storage of the transmitter within its synaptic vesicles. A classic example is the drug reserpine. It depletes monoamine synapses of their transmitters by interfering with their intracellular storage, leading to a profound reduction in neurotransmission.
And what about the metabolism of neurotransmitters within the presynaptic terminal itself? Can drugs influence that step?
Absolutely. Blockade of transmitter metabolism inside the nerve terminal can actually lead to an accumulation of neurotransmitter concentrations. And this has been shown to increase the amount of transmitter released per impulse when the neuron fires. Drugs can also directly alter the release of transmitters. For example, the powerful stimulant amphetamine profoundly induces the release of catecholamines, like dopamine and norepinephrine, from adrenergic synapses. Capsaicin, the chemical responsible for the heat in chili peppers, causes a rapid release of the peptide substance P from sensory neurons. Conversely, tetanus toxin, the infamous bacterial toxin causing severe muscle spasms, exerts its deadly effect by completely blocking the release of inhibitory transmitters in the spinal cord.
Wow. So drugs can either dramatically boost or completely shut down neurotransmitter release. And once the neurotransmitter is released into the synaptic cleft, how is its action normally terminated, and can drugs influence that step?
Right. Once a CNS transmitter is released, its action is rapidly terminated by one of two primary mechanisms, either by reuptake back into the presynaptic terminal or into surrounding glial cells, or by enzymatic degradation within the cleft. For the vast majority of neurotransmitters, reuptake mechanisms are crucial for maintaining signal precision and limiting the duration of action. Cocaine, for instance, is a powerful example. It potently blocks the reuptake of catecholamines, particularly dopamine and norepinephrine, at adrenergic synapses. This blockade greatly potentiates and prolongs the action of these amines, explaining its stimulant and euphoric effects.
Okay, reuptake is key for many. And the other main way, enzymatic degradation. How does that work, and what are the drug targets there?
Acetylcholine is a prime example of a neurotransmitter whose action is primarily terminated by rapid enzymatic degradation, not reuptake. Acetylcholinesterase, an enzyme highly concentrated in the synaptic cleft, rapidly breaks down acetylcholine. Therefore, drugs known as anticholinesterases, which inhibit this enzyme, prolong the action of acetylcholine by preventing its breakdown. Interestingly, for many of the numerous CNS peptides, no conventional uptake mechanism has been conclusively identified, and it's still not entirely clear whether specific enzymatic degradation is the primary mechanism terminating their action in the synapse. That remains an active area of research.
Okay, turning now to the postsynaptic side, the receiving neuron. Here, the primary site of drug action is often the neurotransmitter receptor itself, right? The molecular lock that the neurotransmitter key fits into. How do drugs precisely interact at this crucial level?
Drugs can act as neurotransmitter agonists, meaning they mimic the action of the natural transmitters by binding to the same receptors and activating them. A widely understood example is opioids, which mimic the action of our body's own endogenous opioid peptides, like enkephalins, by binding to and activating opioid receptors, thereby producing pain relief and euphoria. Alternatively, and this is a very common mechanism for CNS drugs, they can act as antagonists, meaning they block receptor function, preventing the natural transmitter from binding or activating the receptor. A striking example here is strychnine's blockade of the receptor for the inhibitory neurotransmitter glycine. This blockade is what directly underlies strychnine's powerful convulsant action, a dramatic illustration of how blocking an inhibitory process results in uncontrolled excitation.
Right. By essentially taking the brake off the system, you effectively accelerate it into overdrive. That's a powerful insight. Can drugs also act directly on the ion channel itself, maybe bypassing the initial receptor binding part?
Yes, they absolutely can. Some drugs bind directly to the ion channel pore of ionotropic receptors, physically blocking the passage of ions. For example, the anesthetic ketamine works by blocking the NMDA subtype of glutamate ionotropic receptors by literally binding inside the ion channel pore, preventing ion flow and disrupting communication. And in the case of metabotropic receptors, drugs can act at any of the steps downstream of the initial receptor binding. Perhaps the clearest example is provided by the methylxanthines, like caffeine. At high concentrations, caffeine elevates the level of cyclic AMP, CMP, not by affecting receptors directly, but by blocking its metabolism by phosphodiesterase enzymes. This effectively prolongs the signaling initiated by other neurotransmitters that utilize CAM10 as a second messenger, contributing to caffeine's stimulant effects.
Okay, now here's where it gets really interesting. Fundamentally challenging a long-held, maybe overly simplistic view. The traditional understanding of the synapse was that it functioned like a simple one-way valve, right? Transmitting information only in a forward direction. But we now have compelling evidence that synapses are far more sophisticated. They can generate signals that feed back onto the presynaptic terminal, essentially modifying and refining transmitter release. This retrograde signaling concept that feels like a significant paradigm shift.
Indeed it is. This two-way communication adds an incredible layer of complexity and fine-tuning. The endocannabinoids are the best documented and most thoroughly studied example of such retrograde signaling. What precisely happens is that postsynaptic activity, the firing or activation of the receiving neuron, triggers the synthesis and release of these endocannabinoids from the postsynaptic neuron itself. These unique molecules then travel backward across the synaptic cleft, activating specific CB1 receptors located on the presynaptic terminal, which belongs to the sending neuron.
So in essence, the receiving neuron is dynamically communicating back to the sending neuron, telling it to perhaps calm down or adjust its message, influencing future communication.
Precisely, yeah. When these presynaptic CB1 receptors are activated by endocannabinoids, they precisely suppress the release of further neurotransmitter from that presynaptic terminal. This suppression can be either transient, lasting for a short period, or remarkably long-lasting, depending on the pattern of neuronal activity. It provides an exquisitely flexible and dynamic feedback mechanism. This is the very pathway through which both endogenous cannabinoids produced by our own brains and exogenous cannabinoids, like those in cannabis, can profoundly affect critical brain functions such as memory formation, cognition, and pain perception, all by modulating presynaptic neurotransmitter release through this elegant retrograde pathway.
Fascinating. Are there other substances that have been proposed to act as retrograde messengers in the CNS beyond endocannabinoids?
Yes. The gas nitric oxide, NO, has long been proposed as a retrograde messenger, primarily because it possesses the unique property of diffusing freely across cell membranes, unlike traditional neurotransmitters. The central nervous system contains a substantial amount of neuronal nitric oxide synthase, NO, an enzyme found within specific classes of neurons. This enzyme is activated by calcium calmodulin, meaning that robust activation of NMDA receptors, which increases intracellular calcium, can lead to the generation of nitric oxide. However, while a clear physiological role for nitric oxide has been established in other systems like vascular smooth muscle, its precise role in synaptic transmission and synaptic plasticity in the CNS is still not fully understood. It remains somewhat controversial.
So the jury's still out on its exact role in brain signaling.
To some extent, yes. Perhaps the strongest and most compelling case for a definitive role of nitric oxide in neuronal signaling in the CNS is its proposed contribution to long-term depression of synaptic transmission in the cerebellum. There, it's thought to mediate a sustained decrease in synaptic strength.
Okay, so connecting this back to our overall mission of achieving mastery, the remarkable specificity of CNS drugs, the ability to achieve targeted therapeutic effects without causing widespread debilitating side effects, this really comes down to two primary interconnected factors. Understanding these allows for unparalleled precision in pharmacology.
Right. It's about designing drugs that are exquisitely precise and hitting their targets within this incredibly complex system. What's the first key factor in achieving that precision? The first factor is neurotransmitter segregation. With only a handful of exceptions, distinct neurotransmitters are released by distinct groups of neurons. Crucially, these transmitters are often segregated into neuronal systems that subserve broadly different CNS functions. This segregation provides neuroscientists and pharmacologists with remarkably precise avenues for analyzing specific CNS functions and for developing targeted treatments. For instance, if you understand that a certain function, like mood regulation or motor control, primarily relies on a specific neurotransmitter system, say dopamine or serotonin, You can then develop drugs that specifically target the neurons that release or respond to that particular transmitter, leaving other systems largely untouched.
That makes profound sense, allowing for highly focused therapies. And what's the second equally crucial factor contributing to this remarkable selectivity?
The second and truly indispensable factor is receptor multiplicity. For a single neurotransmitter, there isn't just one type of receptor molecule. Instead, there's often a remarkable multiplicity of distinct receptor subtypes. Take serotonin, for example. There are at least 14 different receptor types, each encoded by a different gene.
14 for just one neurotransmitter. Wow.
Exactly. And what's more, these different receptor subtypes often exhibit differential distributions throughout the CNS, meaning they're located in different brain regions or on different cell types. This molecular diversity is incredibly powerful because it enables drug developers to design highly selective drugs that target a particular receptor subtype in specific brain regions. This allows for the development of therapies that are far more precise, targeting particular CNS functions while minimizing unwanted side effects by avoiding other receptor subtypes or regions. This is the essence of sophisticated drug design, really hitting the right target in the right place.
Okay, let's unpack this grand organizational scheme of the brain then. When we zoom out from individual neurons and their intricate synapses, the brain's neuronal systems seem to organize into two broad, yet remarkably distinct categories. Each category serves a different fundamental purpose and uses a different communication style, right? Like two completely different types of communication networks.
Indeed. That's a good way to think about it. The first category comprises the hierarchical systems. These are the pathways directly involved in sensory perception, everything from vision and hearing to touch and taste and in motor control, encompassing all our movements, from simple reflexes to complex voluntary actions. These are, in essence, the hardwired pathways, absolutely critical for immediate, precise, and highly localized functions.
So we can think of these as the brain's equivalent of high-speed, dedicated fiber optic cables, designed for rapid, precise data transmission.
Exactly. They are generally characterized by clearly delineated pathways composed of large, heavily myelinated fibers. Myelination, as we discussed, allows them to conduct action potentials incredibly rapidly, often exceeding 50 meters per second. The information flow within these systems is typically phasic, occurring in rapid bursts of action potentials, conveying discrete packets of information. In sensory systems, for example, information is processed sequentially, passing through and being integrated at each relay nucleus on its way up to the cerebral cortex. This serial nature means that a lesion or damage at any single link in these critical pathways can completely incapacitate the entire system, underscoring their precise, non-redundant nature.
Okay, critical, but without much functional redundancy. What specific types of cells make up these hierarchical systems? What are the key players?
Within each nucleus and in the cortical layers of these systems, there are two primary types of cells. First, the relay or projection neurons. These are the workhorses forming the long-distance interconnecting pathways, transmitting signals over vast distances within the brain. They typically have relatively large cell bodies and long axons. However, they also emit small collaterals that synapse onto local interneurons within the same area. These projection neurons are predominantly excitatory, primarily using glutamate as their neurotransmitter, and their synaptic influences, largely involving inotropic receptors, are very short-lived and precise.
And the other crucial cell type, the ones that help refine these signals locally.
The second type is the local circuit neurons, more commonly referred to as interneurons. These are typically smaller than projection neurons, and their axons arborized branch out only in their immediate vicinity. They stay local within a specific nucleus or cortical area. The vast majority of these interneurons are inhibitory, releasing either GABA or glycine as their neurotransmitters. They synapse primarily on the cell body of the projection neurons, but can also connect to their dendrites and even to other interneurons, providing crucial local regulation and control.
In figure 21-5 in the source material, it beautifully illustrates some common types of circuitry these neurons form, doesn't it? Showing their incredible integration capabilities.
Indeed. You'll observe recurrent feedback pathways, where a neuron's output actually feeds back to regulate itself, or previous stages, a powerful self-regulatory mechanism. There are also feed-forward pathways, where one neuron excites another, which then, in turn, excites an inhibitory interneuron that then inhibits a third neuron, a complex way to sculpt information flow. A special and highly important class of local circuit neurons in the spinal cord also forms axoaxonic synapses connections directly on the terminals of sensory axons. This unique arrangement mediates presynaptic inhibition, allowing precise control over sensory input at its very point of entry.
So what are the broader pharmacological implications here? Given the precision and the limited number of key transmitters used in these hierarchical systems, how does that impact drug effects?
Well, despite the immense variety and complexity of these synaptic connections, the fact that a relatively limited number of primary transmitters are used, primarily glutamate for excitation and GABA and glycine for inhibition, means that any major pharmacological manipulation of these systems will have a profound, often widespread effect on the overall excitability of the CNS. For instance, if you selectively block GABA receptors with a drug like picrotoxin, which specifically targets GABA's inhibitory action, the result is generalized convulsions. Thus, while the drug's action is molecularly specific to the GABA receptor, the overall functional effect appears quite nonspecific because GABA-mediated synaptic inhibition is so widely and fundamentally utilized throughout the brain's entire hierarchical system. It demonstrates how a precise molecular action can have a global behavioral consequence.
Okay, that makes sense. Now let's unpack the second grand category of neuronal systems, the nonspecific or diffuse neuronal systems. These operate in a fundamentally different way from the precise hardwired hierarchical systems, right? Instead of conveying specific information, these systems, which use many other neurotransmitters, including the monoamines like dopamine, norepinephrine, serotonin, and also acetylcholine, they seem designed to provide global modulation throughout vast areas of the brain, more like setting the atmospheric conditions, influencing the entire landscape, rather than being precise data lines.
That's a very apt description, yes. What's truly fascinating here is their unique anatomical structure and origin, beautifully illustrated in Figure 21-6. Unlike the widespread distribution of hierarchical neurons, the cell bodies of these diffuse systems are produced by only a remarkably limited number of neurons. These cell bodies are typically located in small, discrete nuclei, often nestled deep within the brainstem. Consider the noradrenergic neurons, for example, those producing norepinephrine. Their cell bodies are found primarily in a compact cell group called the locus coeruleus, containing only about 12,000 such neurons on each side of the human brain.
Only 12,000 for the whole brain.
On each side, yes. Similarly, most serotonin neurons originate from the midline referent nuclei, so an incredibly small number of originating cells.
Yet they manage to have this truly massive, widespread reach across the entire brain. That's a testament to their efficiency, or maybe their design.
Exactly. From these limited nuclei, their axons project widely and incredibly diffusely throughout virtually the entire brain and spinal cord. Their characteristics are quite distinct from hierarchical pathways. Their axons are typically fine and unmyelinated, meaning they conduct signals very slowly, around 0.5 meters per second, a stark contrast to the rapid conduction of hierarchical systems. Furthermore, these axons branch repeatedly and exhibit extraordinary divergence. A single neuron from one of these diffuse systems can innervate multiple functionally different parts of the CNS, synapsing onto and broadly modulating neurons within the hierarchical systems.
That's an astonishing level of divergence. One single neuron influencing so many different, often functionally distinct areas of the brain simultaneously. It's mind-boggling.
It really is. And in the neocortex, these fibers exhibit a tangential organization, meaning they can simultaneously influence very large areas of the cortex in a coordinated manner. Additionally, most neurotransmitters utilized by these diffuse neuronal systems, like norepinephrine, predominantly act on metabotropic receptors, initiating those longer-lasting synaptic effects we discussed earlier. This characteristic reinforces their role as modulators, not rapid information conveyors, setting a tone rather than transmitting discrete messages.
So what are the broader implications of these global modulators for how our brain functions and how drugs affect? What's the big picture takeaway?
Well, based on these observations, it becomes clear that the monoamine systems, for instance, aren't conveying precise, topographically specific types of information. They're not saying this pixel moved here. Instead, they affect vast areas of the CNS simultaneously and in a rather uniform way. Therefore, it's not surprising that these systems have been implicated in global brain functions, such as meticulously regulating our sleeping and waking cycles, our overall attention levels, appetite, and emotional states. They essentially set the overall tone or fundamental state of the brain rather than transmitting specific sensory or motor commands. This understanding is crucial for comprehending the effects of many psychoactive drugs. Now, to truly achieve mastery in CNS pharmacology, it's essential to understand how scientists rigorously identify a chemical as a neurotransmitter. What's fascinating here is the sheer stringency of the criteria required. This process has historically been much harder for central synapses compared to peripheral ones, primarily due to the immense complexity and relative inaccessibility of the brain. However, scientists have meticulously established strict criteria that a substance absolutely must meet to be considered an authentic neurotransmitter.
Okay, so it's not enough to just find a chemical floating around in the brain and assume it's a transmitter. There's a proper scientific burden of proof. What are these strict criteria?
First, there's the localization criterion. A suspected transmitter must be physically present and reside within the presynaptic terminal of the specific neural pathway of interest. You have to show it's there, right where the signal is sent. Second, the release criterion. The suspected transmitter must be verifiably released from a neuron in response to neuronal activity, and critically, this release must occur in a calcium-dependent manner. This confirms it's actively involved in the dynamic process of communication, not just a bystander.
Okay, localization and release. What's the third final criterion that really clinches its identity as a neurotransmitter?
Third, and equally crucial, is the synaptic mimicry criterion. The application of the candidate substance, when introduced experimentally to the synapse, must produce a postsynaptic response that directly and faithfully mimics the action of the natural transmitter released by physiological nerve stimulation. And, crucially, a selective antagonist, when applied, must be able to block that response. This provides compelling evidence that the substance is indeed the natural chemical messenger. Using these rigorous criteria, scientists have identified a range of agents systematically summarized in Table 21-2 in KATZUNGs. We'll now delve into some of the most important of these established neurotransmitters.
Great, let's get into the specifics.
Let's begin our deep dive into specific neurotransmitters with the amino acid neurotransmitters. These truly serve as the fast architects of brain communication. The most prominent ones for pharmacologists fall into two crucial categories: the acidic amino acid, glutamate, and the neutral amino acids, glycine and GABA. These compounds are present in remarkably high concentrations throughout the entire CNS and are extraordinarily potent modifiers of neuronal excitability, fundamentally shaping how our brains process information.
And among them, glutamate is widely recognized as the primary excitatory neurotransmitter, right? The brain's main on switch.
Yes, glutamate is indispensable. It mediates almost all excitatory synaptic transmission throughout the brain. It's found in incredibly high concentrations, around 100 millimolar within excitatory synaptic vesicles, ready for release. When an action potential arrives, glutamate is released into the synaptic cleft through that precise calcium-dependent exocytosis process. Once released, it acts on its specific postsynaptic glutamate receptors and is then very rapidly cleared from the synaptic cleft by specialized glutamate transporters located on surrounding glial cells, as depicted beautifully in Figure 21-7. This rapid clearance ensures signal precision and prevents overstimulation.
That's an efficient removal system. But how does the brain ensure a fresh, continuous supply of glutamate for all this constant excitatory signaling? It must be a tightly regulated cycle, surely.
It absolutely is a marvel of metabolic efficiency. In the glial cells, the cleared glutamate is efficiently converted into glutamine by an enzyme called glutamine synthetase. This glutamine, which is non-neuroactive, is then released from the glia, taken up by the presynaptic nerve terminal, and converted back into glutamate by the enzyme glutinase. Finally, the high concentration of glutamate within synaptic vesicles, ready for the next release, is actively achieved by the vesicular glutamate transporter, VGLUT, which meticulously loads it into the vesicles. So, yes, incredibly efficient, closed-loop recycling system.
Brilliant. Okay, you mentioned that glutamate acts on both inotropic and metabotropic receptors. Let's truly go deep into the inotropic ones first, as they seem central to so much rapid brain function.
Right. The ionotropic glutamate receptors are subdivided into three main classes, named after selective synthetic agonists that helped identify them: AMPA, kainate, and NMDA. All of them are heterotetramers, composed of 4 protein subunits. AMPA receptors, for instance, are ubiquitous, found on virtually all neurons throughout the CNS. They are responsible for the vast majority of fast excitatory synaptic transmission. They're typically assembled from various combinations of 4 subunits, GluA1 through GluA4. The majority of AMPA receptors contain the GluA2 subunit. The presence of this GluA2 subunit is critically important because it makes these receptors primarily permeable to sodium and potassium ions, but crucially, not to calcium ions. However, a fascinating detail for mastery. Some AMPA receptors, particularly those on inhibitory interneurons, lack the GluA2 subunit and as a result are permeable to calcium. This subtle molecular difference has profound implications.
Wow. So a tiny molecular difference in just one subunit dictates whether calcium can flow through, which then impacts the entire signaling of that specific neuron. What about kainate receptors? How do they differ?
Kainate receptors are also ionotropic glutamate receptors, but they're not as uniformly distributed as AMPA receptors. They're expressed at high levels in specific regions like the hippocampus, cerebellum, and spinal cord. They're formed from various subunit combinations of gluK1 through gluK5. Interestingly, gluK4 and gluK5 can't form functional channels alone, but their presence significantly changes the receptor's affinity for glutamate and its kinetics. Similar to AMP receptors, kainate receptors are permeable to sodium and potassium, and in some specific subunit combinations, also permeable to calcium. A clinically relevant connection here is domoic acid, a powerful toxin from certain algae concentrated in shellfish. It's a potent agonist at both kainate and AMPA receptors, and its consumption has caused severe neurological illness, seizures, memory loss.
Right, I remember hearing about that. Okay, and then there are the NMDA receptors. These are quite famous, often implicated in learning and memory, aren't they? They seem to have more complex gating mechanism.
Yes. NMDA receptors are as ubiquitous as AMPA receptors, present on essentially all CNS neurons. All functional NMDA receptors absolutely require the presence of the GLUN1 subunit, along with one or two GLUN2 subunits, GLUN2AD. Unlike AMPA and kainate receptors, all NMDA receptors are highly permeable to calcium as well as sodium and potassium. This calcium influx is an incredibly significant aspect of their function.
And this complex gating you mentioned,
Yeah.
how exactly does it work? It's not just about glutamate binding, is it? There's more to it.
Absolutely not. Their function is controlled in several intriguing and highly regulated ways, making them molecular coincidence detectors. First, in addition to glutamate binding, the channel also requires the binding of glycine to a separate, distinct site for the channel to fully open. The precise physiological regulation of glycine at this site is still an active area of research, though its role as a coagonist is clear. Second, and crucially important for understanding their role in learning, while AMPA and kainate receptors can open at the neuron's resting membrane potential, NMDA receptors typically do not. This is because at resting potential, the NMDA receptor pore is physically blocked by extracellular magnesium ions, Mg2+. Only when the neuron is strongly depolarized, either by intense repetitive activation of the synapse itself through AMP receptors or by simultaneous activation of neighboring synapses, only then is this magnesium expelled from the pore, allowing the channel to finally open and ions to flow.
So it's truly like a highly intelligent gate. It needs both a specific chemical key, glutamate and glycine, and an electrical jolt, that depolarization, to fully open. That's an incredible level of molecular control.
Exactly. This tripartite requirement means NMDA receptors effectively sense strong, sustained synaptic activity, and the subsequent rise in intracellular calcium that accompanies NMDA receptor channel opening is incredibly significant. This is a pivotal aha moment for understanding how our brains learn. This calcium influx triggers a cascade of intracellular signaling events, leading to a long-lasting enhancement in synaptic strength, a phenomenon known as long-term potentiation, or LTP.
LTP.
Right.
The cellular basis of learning.
Precisely. This enhancement of synaptic strength can endure for many hours or even days, and is widely accepted as a fundamental cellular and molecular mechanism underlying learning and memory formation. This is how sustained activity literally hardwires new connections, forming the very foundation of how you acquire and retain information. So when we talk about brain-boosting drugs or understanding learning disorders, we're often looking at ways to fine-tune this calcium influx through NMDA receptors.
That's truly groundbreaking, connecting molecular mechanisms directly to complex cognitive functions like memory. Now, what about the metabotropic glutamate receptors, the MGLRs? How do they differ in their action from the ionotropic ones?
The metabotropic glutamate receptors, MGLURs, are, as their name suggests, G-protein-coupled receptors. They act indirectly on ion channels and other cellular processes via G-proteins. There are 8 identified subtypes, systematically divided into 3 major groups. Group I receptors, MGLUR, MGLUR55, are typically located postsynaptically and, upon activation, trigger the phospholipase C pathway, leading to the release of intracellular calcium from internal stores, initiating a slower modulatory response. In contrast, Group 2, MGLUR2, MGLUR3, and Group 3, MGLUR4, MGLUR6, MGLUR7, and MGLUR8 receptors are predominantly located on presynaptic nerve terminals. They function as inhibitory autoreceptors or heteroreceptors.
Okay, so these presynaptic MGLRs would act as a sophisticated negative feedback loop then, inhibiting further glutamate release, preventing overstimulation, like a governor on the system.
Precisely. Activation of these presynaptic receptors inhibits presynaptic calcium channels, which in turn profoundly inhibits transmission neurotransmitter release. These inhibitory autoreceptors are only activated when the concentration of glutamate in the synaptic cleft rises to sufficiently high levels, usually during intense or repetitive stimulation, acting as that governor on excitation. Their activation also leads to the inhibition of adenylate cyclase, decreasing the generation cyclic AMP or CAMP, further modulating the neuronal response and providing another layer of fine-tuned control over glutamate signaling. Okay, now let's turn our attention to GABA and glycine, the central nervous system's primary inhibitory neurotransmitters. Both of these are crucial for balancing excitation, preventing runaway neuronal activity, and they're typically released from local interneurons. While interneurons releasing primarily glycine are largely restricted to the spinal cord and brainstem, interneurons releasing GABA are ubiquitous, present throughout the entire CNS, including the spinal cord, cerebellum, cortex, everywhere. This broad distribution makes GABA particularly important.
So glycine is more localized, while GABA exerts widespread inhibitory control. What kind of receptors do they act on, and how do their mechanisms lead to inhibition?
Glycine receptors are pentameric structures composed of 5 protein subunits, and they are selectively permeable to chloride ions, Cl. When glycine binds, these channels open, allowing chloride to flow into the cell, which leads to hyperpolarization or stabilization of the membrane potential, thus inhibiting the neuron. A striking and clinically significant fact, strychnine, that potent spinal cord convulsant we mentioned earlier, used in some rat poisons unfortunately, selectively and powerfully blocks glycine receptors. This provides a vivid, dramatic demonstration of how blocking inhibition, specifically glycine-mediated inhibition in the spinal cord, leads to widespread, uncontrolled excitation, manifesting as severe spasms and convulsions. It really underscores the vital importance of this inhibitory balance.
It's a very clear, powerful illustration. The critical importance of inhibitory balance. And GABA, which you said is widespread. How are its receptors classified, and what are their distinct roles?
GABA receptors are critically important and are divided into 2 main types, GABA and GABAB. Interestingly, inhibitory postsynaptic potentials, IPSPs, in many areas of the brain actually have both a fast and a slow component. The rapid transient component of these IPSPs is mediated by GABA receptors, while the slower, more prolonged component is mediated by GABA receptors. This difference in kinetics stems directly from how these distinct receptor types are coupled to ion channels.
Okay, so GABA is the fast one. How does it work at the molecular level to achieve this rapid inhibition?
GABA receptors are ionotropic receptors. Like glycine receptors, they are pentameric structures, forming a chloride ion channel selectively permeable to chloride ions. When GABA binds, the channel opens rapidly, allowing chloride influx, which hyperpolarizes or shunts the membrane, causing fast inhibition. These receptors are selectively and powerfully inhibited by compounds like picrotoxin and bicuculane, both of which, similar to strychnine, cause generalized convulsions by blocking this crucial inhibitory pathway. A vast number of different subunits have been cloned for GABA receptors, and their varied combinations account for the immense diversity in their pharmacology and precise locations. This remarkable diversity makes them absolutely key targets for a wide array of clinically useful agents, like benzodiazepines, think Valium or Xanax, and barbiturates like phenobarbital. These exert their therapeutic effects precisely by enhancing GABA receptor function, making the brain more inhibited, less excitable.
Right, that makes sense why they're used for anxiety or seizures. And then there's the GABA receptor, mediating the slower, more prolonged component of inhibition. How does it achieve its effects, and what's its clinical relevance?
GABA receptors are metapopic receptors. They're selectively activated by baclofen, an important antispastic drug used to relieve muscle rigidity. These receptors are coupled to G proteins that, depending on their precise cellular location, either inhibit presynaptic calcium channels or activate postsynaptic potassium channels. The GABA component of the inhibitory postsynaptic potential is primarily due to a selective increase in potassium conductance. Potassium ions flow out of the cell, making the inside more negative. This IPSP is notably long-lasting and slow, precisely because the coupling of GABA receptor activation to potassium channel opening is indirect, mediated by a G protein, and thus inherently delayed compared to direct ionotropic action.
Okay, indirect coupling leads to slower, longer effects. And where are these GABA receptors typically localized, and do they have additional functions beyond activating potassium channels?
GABA receptors are often localized to the perisynaptic region, meaning just outside the immediate synaptic cleft. They seem to require a slight spillover of GABA from the cleft to be activated. This implies they respond to more robust or sustained GABA release. GABA receptors are also prominently present on the axon terminals of many excitatory and inhibitory synapses. In these presynaptic locations, GABA spillover onto these GABA receptors inhibits transmitter release by precisely inhibiting presynaptic calcium channels, effectively putting a brake on signal transmission from that terminal. Beyond their coupling to ion channels, GABA receptors also inhibit adenylyl cyclase, leading to a decrease in CAMP generation. This provides yet another layer of inhibitory control, influencing a wide array of neuronal processes, illustrating their widespread modulatory role. Let's talk about acetylcholine, Natus. It holds a truly special place in neuroscience history because it was the very first compound pharmacologically identified as a bona fide neurotransmitter in the CNS. Pioneering work by Eccles and his collaborators in the early 1950s provided compelling evidence. They show that the specific excitation of spinal cord Renshaw cells, a type of interneuron by recurrent axon collaterals from spinal motor neurons, was specifically and potently blocked by nicotinic antagonists. Furthermore, those Renshaw cells themselves were exquisitely sensitive to the application of nicotinic agonists. This was direct, unequivocal proof of ACE's role in central communication.
Definitive evidence right there in the spinal cord. And what types of receptors does acetylcholine act on in the complex environment of the CNS? Is it just nicotinic?
In the CNS, we find ionotropic nicotinic receptors, yes. Though it's important to note their subunits are entirely distinct from the nicotinic channels found at the neuromuscular junction, which control muscle contraction. These central nicotinic receptors are widely distributed throughout the brain and spinal cord, mediating rapid excitatory effects. However, and this is crucial for mastery, the vast majority of central nervous system responses to acetylcholine are actually mediated by a large and diverse family of G protein-coupled muscarinic receptors, not nicotinic ones.
Muscarinic, okay. And what are the different, often complex actions mediated by these muscarinic receptors? Do they all cause excitation, or is it more varied?
Not at all. Their actions are quite varied and nuanced. At a few specific sites, acetylcholine causes a slow inhibition of the neuron by activating the M2 subtype of muscarinic receptor, which then opens specific potassium channels, leading to hyperpolarization. But a far more widespread and characteristic muscarinic action in response to acetylcholine is actually a slow excitation. This excitation, in some cases, is mediated by M1 receptors. What's particularly unusual and noteworthy about this M1 muscarinic excitation is that acetylcholine produces it by decreasing the membrane permeability to potassium.
Wait, decreasing permeability? That's the opposite of conventional transmitter action, isn't it? Usually they increase permeability.
Exactly. It's blocking a brake to cause excitation. This unique mechanism highlights the complexity of neuromodulation.
That's quite counterintuitive. Fascinating. Where are these cholinergic neurons located in the brain, and what essential functions are they involved in?
8 major CNS nuclei containing acetylcholine neurons have been precisely characterized, and they possess diffuse projections, similar to the monoamine systems we'll discuss. These include neurons in the neostriatum, the medial septal nucleus, and the reticular formation. These widespread cholinergic pathways appear to play a crucial role in various cognitive functions, most notably memory. From a clinical perspective, a profound loss of these cholinergic neurons, particularly in the basal forebrain, is consistently associated with presenile dementia of the Alzheimer type. This highlights their profound importance in healthy brain aging and cognitive function, making them a significant target for understanding and treating neurodegenerative diseases. Okay, let's pivot now to the monoamine neurotransmitters. This crucial group includes the catecholamines, dopamine, and norepinephrine, and 5-hydroxytryptamine, more commonly known as serotonin. We also include histamine here due to its shared characteristics and broad modulatory roles with these monoamines. Although these compounds are present in relatively small amounts within the CNS compared to amino acids, they exert wide-ranging and profound effects. And they are, critically, the primary site of action for many clinically important psychoactive drugs. For example, the powerful CNS stimulants, cocaine and amphetamine, primarily act at catecholamine synapses. Cocaine potently blocks the reuptake of dopamine and norepinephrine, dramatically prolonging their action, while amphetamines cause presynaptic terminals to release these transmitters directly.
Right. So these monoamines are fundamental to mood, motivation, arousal. And drugs targeting them modulate their availability or release. Let's delve into dopamine first. What are its major pathways in the brain and its general actions?
The major pathways containing dopamine are incredibly significant. One prominent pathway links the substantia nigra to the neostriatum. This pathway is profoundly implicated in Parkinson's disease and is the primary target for the therapeutic action of levodopa. The other major pathway links the ventral tegmental region to limbic structures, particularly the limbic cortex. And this pathway is thought to be intimately associated with the therapeutic action of antipsychotic drugs used to treat conditions like schizophrenia. Additionally, dopamine-containing neurons in the ventral hypothalamus play a crucial role in regulating pituitary function, influencing hormone release.
Okay, very distinct pathways linked to very different functions and diseases. And what about the specific types of dopamine receptors that mediate these diverse effects?
5 distinct dopamine receptors have been identified, falling into 2 main categories: the D1-like receptors, D1, D5, and the D2-like receptors, D2, D3, D4. It's important to note that all 5 identified dopamine receptors are metabotropic. They're G-protein-coupled receptors acting indirectly via second messengers. When applied to CNS neurons, dopamine generally exerts a slow inhibitory action. This action has been best characterized on the dopamine-containing neurons of the substantia nigra, where D2 receptor activation opens potassium channels via its G protein, leading to hyperpolarization and inhibition. However, dopamine also plays complex excitatory and modulatory roles depending on the receptor subtype and neuronal context.
Okay. Moving on to norepinephrine, or NE. Where are its key neurons located, and how does it act in the CNS, especially given its known role in attention and arousal?
Most noradrenergic neurons, those producing and releasing norepinephrine, are primarily located in 2 discrete nuclei: the locus coeruleus in the brainstem, or the lateral tegmental area of the reticular formation. Although the density of noradrenergic fibers innervating various brain regions differs considerably, most regions of the CNS receive a diffuse noradrenergic input, underscoring its widespread modulatory role. All identified noradrenergic receptor subtypes are metabotropic. When norepinephrine is applied to neurons, it can hyperpolarize them by increasing potassium conductance. This inhibitory effect is typically mediated by receptors and has been characterized most thoroughly on the neurons of the locus coeruleus itself, providing a self-regulatory feedback mechanism.
But doesn't norepinephrine also have a critical excitatory or facilitatory role in many brain functions, especially those linked to attention? How does that work?
It absolutely does, and this is a key nuance for understanding its pharmacology. In many regions of the CNS, norepinephrine actually enhances excitatory inputs through both indirect and direct mechanisms. The indirect mechanism involves disinhibition. Norepinephrine inhibits inhibitory local circuit neurons, effectively taking the brakes off other excitatory signals, allowing them to propagate more effectively.
Inhibiting the inhibitors. Clever.
Exactly. The direct mechanism involved the blockade of potassium conductances that would normally slow down neuronal discharge, thereby making neurons more excitable. Depending on the specific neuron and brain region, these facilitatory effects are mediated by either ECO1 or Avest receptors. This overall facilitation of excitatory synaptic transmission aligns perfectly with norepinephrine's well-established roles in behavioral processes like attention, arousal, vigilance, and the general fight-or-flight response. Next up is 5-hydroxytryptamine, 5-HT, universally known as serotonin. Most serotonin pathways originate from neurons located in the midline raphe nuclei situated in the pons and upper brainstem. Serotonin is contained within unmyelinated fibers that project widely and diffusely, innervating most regions of the CNS, although the density of innervation can vary considerably. This widespread projection underlies its broad influence.
And serotonin is famous for having a truly remarkable number of receptor subtypes, isn't it? Making it a really complex target for drug development.
That's correct. Serotonin acts on an astonishing diversity of over a dozen receptor subtypes, making it one of the most complex neurotransmitter systems. With the sole notable exception of the 5-HT3 receptor, all of these identified receptors are metabotropic. The inotropic 5-HT3 receptor, while distinct, exerts a rapid excitatory action, but only at a very limited number of sites in the CNS. In contrast, in most areas of the CNS, serotonin actually has a strong inhibitory action. This is primarily mediated by 5-HT1A receptors and is consistently associated with membrane hyperpolarization caused by an increase in potassium conductance. Interestingly, for mastery, 5-HT1A receptors and GABAB receptors activate the same population of potassium channels, highlighting an intriguing convergence in inhibitory mechanisms.
Okay, so it's predominantly inhibitory, but you mentioned it can also have excitatory actions. How does that dual role play out? It seems complicated.
Yes, it's one of its complexities. Some specific cell types in the brain are slowly excited by serotonin, primarily due to its blockade of certain potassium channels via 5-HT2 or 5-HT4 receptors. What's truly fascinating is that both excitatory and inhibitory actions can occur on the same neuron depending on the precise complement of serotonin receptors expressed on that particular cell. Given the remarkably broad roles of serotonin in virtually all brain functions, perception, mood, anxiety, pain, sleep, appetite, temperature, neuroendocrine control, aggression.
Wow, basically everything.
Pretty much. And coupled with the rich molecular diversity of its receptors, it's entirely unsurprising that a vast number of therapeutic agents currently target or modulate the 5-HT system to treat a wide range of neuropsychiatric conditions. Finally, for our discussion of monoamines, let's touch on histamine. In the central nervous system, histamine is exclusively synthesized and released by neurons located in a very specific nucleus, the tuberomammillary nucleus, or TMN, situated in the posterior hypothalamus. From this discrete origin, these histaminergic neurons project widely throughout the entire brain and spinal cord, where they modulate crucial physiological functions such as arousal, attention, feeding behavior, and memory.
And what kind of receptors does histamine interact with to mediate these widespread effects? Are they also diverse?
There are 4 identified histamine receptors, H1 through H4, and all of them are metabotropic G protein-coupled receptors. From a clinical perspective, centrally acting antihistamines are perhaps most widely known for their sedative properties. This sedation is a common side effect of many drugs, including certain older tricyclic antidepressants and antipsychotics, because they often possess a significant antagonistic effect on H1 receptors in the brain. Understanding this antagonism helps explain why some medications can make you drowsy, linking a specific molecular interaction to a common behavioral effect. Okay, beyond the classic small molecule neurotransmitters, a great many CNS peptides, often called neuropeptides, have been discovered. These produce dramatic and diverse effects on both animal behavior and the activity of individual neurons. In many cases, these were initially identified as peptide hormones in the periphery outside the brain and were only later found to also act as neurotransmitters within the CNS. Because most were initially named based on their peripheral functions, their names are often quite unrelated to their specific, sometimes unexpected CNS functions. This can be a point of confusion, but also a fascinating insight into evolution reusing molecules.
So their names can be a bit misleading in the context of their brain function. Can you give us a few examples of these important neuropeptides? What are some key ones?
The precise neural pathways for many have been meticulously mapped using advanced immunohistochemical techniques. Prominent examples include various opioid peptides like enkephalins and endorphins, our body's natural painkillers, also neurotensin, substance P, somatostatin, cholecystokinin, CCK, vasoactive intestinal polypeptide, VIP, neuropeptide Y, NPY, and thyrotropin-releasing hormone, TRH, among many others. It's a rapidly expanding field.
Do these neuropeptides behave identically to the classic small molecule neurotransmitters we've been discussing, like glutamate or dopamine, or are there key distinctions in how they're made, packaged, and act?
That's a crucial question for deep understanding, and no, they do not behave identically. They have some very important distinctions. Unlike classic neurotransmitters, typically packaged in small synaptic vesicles, neuropeptides are generally packaged in larger, denser core vesicles. Also, similar to what we see in the peripheral autonomic nervous system, peptides often coexist with a conventional non-peptide transmitter in the same neuron. But their release can be independently regulated.
Independently regulated, meaning the neuron can choose which message to send.
To some extent, yes. Depending on the intensity or pattern of stimulation, the neuron can preferentially release different chemical messengers, allowing for more nuanced communication.
Fascinating. And once released, how do they act on other neurons? Do they also have different patterns of influence compared to the small molecules?
Released neuropeptides may act locally, much like classic transmitters, but they also possess the capacity to diffuse longer distances and bind to receptors far from their release site. This contributes to more diffuse, widespread, and often long-lasting effects. The majority of neuropeptide receptors are metabotropic, and critically, similar to monoamines, they primarily serve modulatory roles in the nervous system. They don't typically mediate rapid, point-to-point information transfer, but rather fine-tune and sculpt the excitability and responsiveness of neuronal circuits over longer timescales. These distinctions highlight their unique and often profound impact.
And what kind of broad functions are these neuropeptides implicated in? Given their modulatory nature and widespread distribution, what do they generally influence?
They are implicated in an incredibly wide range of CNS functions, far beyond simple signaling. This includes crucial roles in reproduction, complex social behaviors, appetite regulation, arousal states, pain perception and modulation, reward pathways, and critical aspects of learning and memory. This broad and often fundamental involvement makes them incredibly active and promising targets for ongoing drug discovery efforts, as modulating these systems could lead to therapies for conditions currently hard to treat.
Can you give us a specific example, maybe one that challenged initial assumptions about how the brain works? How studying a neuropeptide illuminated brain function?
A very good example comes from studies on substance P and its association with sensory fibers. Substance P is contained within and released from small, unmyelinated primary sensory neurons found in the spinal cord and brainstem. When released, it causes a slow, excitatory postsynaptic potential in its target neurons. Because these specific sensory fibers are known to transmit noxious stimuli pain signals, it was initially hypothesized that substance P was the primary, perhaps even sole, neurotransmitter for pain.
That seems logical initially.
It did. However, it's quite surprising to learn that although substance P receptor antagonists can indeed modify responses to certain types of pain, they do not completely block the pain response.
Ah, so it's involved, but clearly not the whole story for pain,
Yeah.
implying a more complex interplay.
Exactly. This discovery led to a crucial refinement in our understanding. Glutamate, which is often co-released with substance P from these very same synapses, is now presumed to play a more important, perhaps dominant role in transmitting pain stimuli. Furthermore, substance P is definitely involved in many other critical CNS functions because it is found in numerous areas of the central nervous system that are anatomically and functionally completely unrelated to classical pain pathways. This powerfully highlights the complexity and often overlapping pleiotropic roles of neurotransmitters and how our understanding evolves as we gather more data. It's rarely simple. Okay, beyond the established classical neurotransmitters and neuropeptides, the central nervous system also utilizes other fascinating signaling substances that continually expand our very definition of neuronal communication. Let's delve into a few of these, starting with the endocannabinoids. These have really revolutionized our understanding of synaptic plasticity.
Ah, right. These are the brain's own natural cannabis-like chemicals, responsible for many of the effects of cannabis itself.
Precisely. The primary psychoactive ingredient in cannabis, delta-9 tetrahydrocannabinol, or 9-THC, exerts its effects on the brain mainly by activating a specific cannabinoid receptor known as CB1. CB1 receptors are remarkably highly expressed in many brain regions, and critically, they are predominantly located on presynaptic terminals, as elegantly depicted in Figure 21-8 in KATZUNGs. Our bodies naturally produce endogenous ligands for these CB1 receptors. These include compounds like anandamide and 2-arachidonylglycerol, or 2-AG. These are collectively referred to as endocannabinoids.
And how do these endogenous cannabinoids work? Are they synthesized and stored in vesicles like traditional neurotransmitters waiting to be released?
No, and that's one of their most unique and defining features, crucial for understanding their modulatory role. Unlike classic neurotransmitters, endocannabinoids are not stored in synaptic vesicles. Instead, they are rapidly synthesized by neurons on demand, typically in response to a significant calcium influx into the postsynaptic neuron, or following robust activation of metabotropic receptors by other neurotransmitters like acetylcholine and glutamate. This on-demand synthesis is absolutely crucial. And in a further profound contrast to classic neurotransmitters, endocannabinoids function primarily as retrograde synaptic messengers.
Retrograde, meaning they signal backward across the synapse, from the receiving neuron back to the sending neuron. That's a truly revolutionary concept in neuroscience, isn't it?
Exactly. They are released from the postsynaptic neurons, the receiving cells, and then travel backward across the synaptic cleft, activating CD1 receptors located on the presynaptic neurons, the sending cells. This activation of presynaptic CB1 receptors then suppresses the release of further neurotransmitter from that presynaptic terminal. This suppression of release can be either transient, providing short-term feedback, or remarkably long-lasting, allowing for more sustained modulation of synaptic strength. Both endogenous cannabinoids and exogenous cannabinoids, like Day9-THC, are believed to profoundly affect memory, cognition, and pain perception through this precise and dynamic retrograde mechanism. It reveals a whole new layer of synaptic communication and control. Another intriguing signaling substance that has expanded our definition is nitric oxide, NO. It's a gas. The central nervous system contains a substantial amount of neuronal nitric oxide synthase, NOS, an enzyme found within certain classes of neurons. This enzyme is activated by calcium calmodulin complexes, meaning that robust activation of NMDA receptors, which increases intracellular calcium, directly results in the generation of nitric oxide.
So, intense glutamate signaling through NMDA receptors can directly lead to NO production. Is its precise role in synaptic transmission and plasticity fully established? Or is it still debated?
Well, While a clear and indispensable physiological role for nitric oxide has been definitively established in other systems, like regulating vascular smooth muscle, its precise role in synaptic transmission and synaptic plasticity in the CNS remains an area of active investigation and, yes, some controversy. Nitric oxide is unique because it diffuses freely across membranes, which initially led to the compelling hypothesis that it acts as a retrograde messenger similar to endocannabinoids. However, this hasn't been demonstrated conclusively across all synapses. Perhaps the strongest and most widely accepted case for a role of nitric oxide in neuronal signaling in the CNS is its proposed contribution to long-term depression of synaptic transmission in the cerebellum. There, it's thought to mediate a sustained decrease in synaptic strength, a form of neuroplasticity essential for motor learning.
Okay, so important, but maybe not as universally acting as initially thought.
Possibly. The research continues. Finally, let's look at purines, a fascinating class of signaling molecules including adenosine, ATP, adenosine triphosphate, UTP, uridine triphosphate, and UDP, uridine diphosphate. Receptors for these purines are found ubiquitously throughout the body, including extensively within the central nervous system. High concentrations of ATP are found in and released from many classical neurotransmitter synaptic vesicles, particularly catecholaminergic ones. Interestingly, ATP may also be released by astrocytes, those glial support cells, to modulate neuronal function, showcasing another layer of glia-neuron interaction.
Astrocytes releasing ATP too. Wow. And what types of receptors does ATP bind to in the CNS to exert its effects?
ATP primarily binds to two major classes of purine receptors. First, the P2X family, which consists of non-selective ligand-gated cation channels, ionotropic receptors allowing rapid ion flow when ATP binds. Second, the P2Y family, which are metabotropic receptors acting indirectly via G proteins and second messengers. While the exact physiological roles for ATP release and purine signaling in general in the CNS are still being actively elucidated, pharmacological studies strongly suggest these receptors are intricately involved in memory formation, regulating wakefulness and appetite control. Furthermore, they may play significant roles in the pathophysiology of multiple neuropsychiatric disorders, making them attractive new targets for drug discovery.
Okay, and then there's adenosine. Many of us are most familiar with that one because of its interaction with caffeine, right? How does adenosine exert its effects in the CNS, especially regarding sleep and wakefulness?
Indeed. Extracellular ATP, once released, is efficiently converted to adenosine by a cascade of ectonucleotidases, enzymes located outside the cell. Adenosine in the CNS acts on two main types of metabotropic receptors, A1 and A2A adenosine receptors. Presynaptic A1 receptors, when activated, inhibit calcium channels and in turn broadly inhibit the release of both amino acid and monoamine transmitters, essentially acting as a general brake on synaptic transmission. Postsynaptic A2A receptors, on the other hand, increase cyclic AMP production and generally have a mildly excitatory effect on neurons, although their primary role is likely modulatory.
So how do these seemingly opposing actions of adenosine receptors relate to sleep and wakefulness? And how does caffeine fit into this picture? It seems a bit paradoxical.
It's a beautiful example of fundamental pharmacology at play. Increasing levels of extracellular adenosine in the CNS directly promote drowsiness and induce sleep, largely through its action on the A2A receptors, which modulate neuronal activity related to arousal. And this is precisely where caffeine, one of the most widely consumed psychoactive substances, enters the scene. Caffeine is a non-selective antagonist at adenosine receptors. By blocking adenosine's action, particularly on the A2A receptors, caffeine effectively disinhibits arousal pathways, thereby promoting wakefulness and reducing feelings of fatigue. It's a classic everyday example of how a common substance exerts its effects by interfering with a fundamental, evolutionarily conserved signaling pathway in the brain, showcasing the power of understanding these molecular interactions. So if we connect this truly deep dive back to the bigger picture, we've embarked on an exhaustive journey through the intricate, multi-layered architecture of the central nervous system. We've navigated from the individual neuron and its essential, often underappreciated, supporting glial cells, meticulously crossing the highly selective blood-brain barrier. We then delved into the pulsating, dynamic world of ion channels and the diverse landscape of neurotransmitter receptors. We meticulously dissected the complex synaptic symphony, getting a profound understanding of how drugs precisely target and modify each step of this communication. We explored the two grand organizational principles of the brain's circuitry, the rapid, precise hierarchical systems and the global, diffuse, modulatory systems, understanding their distinct roles. And finally, we unpack the diverse and fascinating family of chemical messengers, from the primary excitants like glutamate and inhibitors like GABA, to the influential monoamines, the complex neuropeptides, and even unexpected players like endocannabinoids and purines. You now possess, I hope, a deep foundational understanding, the kind of nuanced insight that truly empowers you to master this information like the elite professionals in this field.
So, what does this all mean for you, armed with this mastery? Consider this provocative thought. The incredible specificity, the molecular diversity, the delicate balance of these CNS neurotransmitter systems. It means that even subtle, precise changes in their function can have profound, far-reaching effects on our perception of reality, our moods, our fundamental behaviors. As we continue to unravel these breathtaking complexities with ever-increasing precision, just imagine the immense potential for even more targeted, exquisitely fine-tuned therapies. These could offer unprecedented control over our mental and emotional states, perhaps even enhancing cognitive capabilities we're only just beginning to understand. As our mastery over these subtle chemical dances grows, what profound ethical considerations might we as a society increasingly face? Something to think about. Thank you for joining us on this truly deep dive into the pharmacology of CNS drugs. Keep exploring, keep questioning, and keep learning.